Welcome back. So we've talked about what translation is and how we use a genetic code and what a codon is and what mutations do. Let's start meeting some of these characters involved. So the main ones we have are ribosomes. This little two circly thing is a ribosome. That is the main apparatus, the kind of scaffolding used to carry out the process of polypeptide synthesis. tRNA, which is this little key-shaped thing, is going to bring in the correct amino acid. It has this anticodon, 3 bases on here, that will base pair with the mRNA and bring in the correct amino acid. Aminoacyl-tRNA synthetases is this part of the key. It fits in a specific tRNA and a specific amino acid, so it makes sure that every tRNA with the specific anticodon has a specific amino acid attached to it. So if this is going to be bringing in this amino acid, it is going to fit into its specific aminoacyl-tRNA synthetase. So there are different aminoacyl-tRNA synthetases for the different tRNAs and amino acid combinations we make. mRNA, which is just represented here as a string of letters, is going to bring the nucleotide sequence from DNA, which can then be decoded to create the protein synthesis. And then there are protein factors that are used in some of the steps of translation that we'll talk about. Ribosomes are made of RNA and protein. In eukaryotes, as you remember, they're either bound to the endoplasmic reticulum or the outer nuclear envelope, if they're bound ribosomes. If they're free ribosomes, they are floating around in the cytoplasm. Both bound and free ribosomes are made of exactly the same thing. The only difference is they are attached or not attached. Free ones can become bound, and bound can become free. Ribosomes in prokaryotes and eukaryotes are a little bit different. Prokaryotic ribosomes are a little bit smaller. There's a little bit difference in their density. But either way, they are made of 2 subunits, a large and a small. Here is a large and a small to make a 70S prokaryotic ribosome. And here's a large and small to make an 80S or eukaryotic ribosome. So these numbers, you can see they don't add together. This isn't like a weight or anything like that. This S on it is telling you what layer it'll sediment. So if you were to take a cell and centrifuge it, you would separate it into different layers. And each of those different layers has a different number assigned to it. So that's what this means, is where you'll find it. It's a mixture of what its shape is and what its size is, what its density is. But you can see if a bacterial ribosome is 70S, it's smaller and less dense than a eukaryotic ribosome. A eukaryotic ribosome is 80S. It's larger and more dense than our prokaryotic ribosome. Bacterial ribosomes are going to be sensitive to different inhibitors of protein synthesis, and they're composed of fewer proteins and smaller and fewer RNA molecules. This ability to have some medicines that can inhibit a bacterial ribosome without inhibiting a eukaryotic ribosome means we can make drugs like streptomycin or tetracycline that will attach to a bacterial ribosome and inhibit them and kill the bacterial infection. But those same drugs will not attach to a eukaryotic ribosome, so they won't attack and kill your own cells. So you can use that difference in bacterial ribosomes to kill an infection without harming the person who is taking the drug. Ribosomes are going to be involved in polypeptide synthesis. They're going to look like a big complex enzyme. They've got more than 50 proteins. They've got lots of different rRNA molecules put together. And a lot of the work that a ribosome is doing is because of the ribosomal RNA in them. It's not only the proteins working, but that combination of proteins and rRNA. There's different sites within the ribosome that are important for protein synthesis. One of them is the mRNA binding site. That is where the mRNA is going to be able to come through, and the mRNA will just move through here, allowing you to add amino acids one codon at a time. Then you've got these three little pill-shaped spots here. The first one is the A site, or the aminoacyl site. This is where the tRNA is going to come in and bring an amino acid. So the tRNA will be interacting with the mRNA, codon to anticodon, and bringing in the amino acid. Then when that correct tRNA clicks in, it's going to shift down, and that amino acid will now be in the P site, or the peptidal site. And a new tRNA will come in to fit in with the codon that's just moved in. So you'll now have two little tRNAs with two little peptides here, and you'll form a peptide bond, or a covalent bond between those two amino acids. Then when you form that covalent bond, it shifts down again. The tRNA that was in the P site is now in the E site, which is exit site, and that tRNA is going to leave. The tRNA that was in the A site moves down to the P site. A new codon is now in the A site. New tRNA comes in, form a new peptide bond, shift down, exit, bring in a new amino acid. Form a peptide bond, shift down, exit again and again, creating a longer and longer peptide chain. Our tRNA is a molecule that it kind of has, conveniently forms this kind of clover leaf like a T shape. It is going to bind to both a specific amino acid. So here it has an amino acid attachment site that will bind with a covalent bond to a specific amino acid. And it has an anticodon, these three little bases of the RNA that will interact with the codon on the mRNA. This bond that holds the tRNA to the amino acid is called an ester bond. It's this covalent bond where you have an oxygen between your carbons. And each tRNA is going to be named after the amino acid it's carrying. So an amino acid that is carrying, a tRNA that's carrying the amino acid alanine is going to be labeled like this: tRNA ALA, ALA for alanine. This is going to kind of fold up into this shape. So these loops kind of fold up together to have this more compact shape that's going to then interact into the ribosome, move into the A site, bringing in that next amino acid. tRNAs that have an amino acid attached to them, like this one, are called aminoacyl-tRNAs. And we call this tRNA as charged. The amino acid we say is activated. The tRNA will recognize the codon in mRNA because its anticodon will base pair. And tRNAs are able to do something called wobble. They're able to recognize more than one codon. So this anticodon will be able to recognize a few different codons. If you remember talking about the degeneracy of the genetic code, that's where that degeneracy comes from, is when you have tRNAs that can bind to multiple codons. So anticodons allow tRNA molecules to recognize the codons in mRNA by complementary base pairing. So this tRNA here has it doesn't show. I thought it said UUU. That would mean it would be able to recognize the AAA codon in the RNA. So mRNA and tRNA will line up to each other. Here is your mRNA carrying its nucleotides, your tRNA carrying its nucleotides. And the way that they line up on the ribosome allows a little bit of wobble, a little bit of flexibility between that third codon of the mRNA and the corresponding part of the anticodon. So this wobble hypothesis lets there be a little bit of wiggle room in that base pairing. That means because of wobble, we don't have to have 64 different tRNAs. You can have fewer tRNAs than the number of codons that you have. So here this actually tells you if there is a U in the codon, it'll be recognized by an A in the anticodon in the first position. The G will be recognized by a C. So here, this codon is AGA. So your tRNA has UCU because A and U base pair and G and C base pair. So that tRNA comes in and is able to interact with that. Before that tRNA can even come in and bring the amino acid, it has to be able to be attached to the correct amino acid. And aminoacyl-tRNA synthetase is a molecule that's going to attach the tRNA and the amino acid. So cells have 20 different aminoacyl-tRNA synthetases for those 20 amino acids that we have. So each amino acid has its unique aminoacyl-tRNA synthetase. And we can name them after, this one is for tyrosine and this one is for valine. There are some cells with some non-traditional amino acids, and they'll have some special tRNAs, some special aminoacyl-tRNA synthetases for those unique non-traditional amino acids. Things like the inosine that we mentioned on the last slide. Aminoacyl-tRNA synthetases will make that tRNA and the amino acid, that covalent bond will be catalyzed using an ester bond, and that will require ATP hydrolysis. So now you have this amino acid, your tRNA, we hydrolyze it, take out this H2O, and this now we have an O ester bond here between the amino acid and the tRNA. Both the anticodon on the 3' end of the tRNA are going to be needed to specify the correct both the anticodon and the 3' end of the tRNA are going to be needed to make sure that the correct amino acid comes in. And when you add the amino acid or the amino acid activation, these synthetases are going to proofread to make sure that you added, in fact, the correct amino acid. And the tRNA that will be able to recognize the appropriate codon in mRNA. The sequence of codons in mRNA will direct the order of amino acids in the polypeptide. So mRNA that was made in the nucleus is going to be in a eukaryotic cell. You may have some snipping of exons and introns and creating your final mRNA and then exporting it to the cytoplasm. And at the 3' end of that message, you have an untranslated sequence. So it's not going to start exactly at the first part of it. You're going to look for a start codon, AUG. And that AUG is going to be bound by the initiator tRNA, which has a UAC on it and is always carrying methionine. And each of these steps is going to require different protein factors or proteins that help set up what's going through these steps. At the 3' end, we also have an untranslated region. So once you have the stop codon, translation stops, but the mRNA can continue past that. This stop codon is going to be generally UAG, UAA, or UGA. We did talk about another stop codon in mitochondria, but in most of our eukaryotic business, we're using these 3 stop codons. So even though you have an untranslated region at the 5' end and an untranslated region at the 3' end, and they can be different lengths, but they're still important for RNA function. And then beyond that 3' end and 5' end, you also have a poly A tail and a 5' cap. Most RNAs in eukaryotes are going to be monocistronic. That means when they are monocistronic, that one mRNA is going to create one polypeptide. In bacteria and archaea, they are a little bit cool. They sometimes can make mRNAs that are polycistronic. If they are polycistronic, that mRNA will be able to create more than one protein. We're going to talk about a couple of operons in bacteria. That means that you have multiple polypeptides created from one mRNA with related functions. So it may be now we're going to be able to make 3 enzymes that we need to be able to digest lactose when lactose is around. That would be a type of polycistronic polypeptide, polycistronic mRNA in bacteria. It'll lead to 3 different polypeptides. And that polycistronic transcription unit is called an operon. Operons are really fascinating. We're going to come back and talk about them, not in this chapter, but in the next chapter, how they work. All right, so we've talked about everything that's involved in the process of making our protein. We haven't actually started making our protein yet. So in our next video, we're going to start exploring how that works. I'll see you then. Bye.